Okay, so picking up where we left off the other day, and after our activity building these two operons, we're going to start with the lac operon, as these two, the lac and the trip operon, are just good examples of two different types of operons. So starting with the lac, first of all, um, the lac operon is it's the set of genes that allows us to break down lactose as an energy source. So without this operon, we would not be able to use lactose as a energy molecule. Preferentially, our body does use glucose, but lactose is a possibility. And so in order to produce the enzyme lactase, we go through a pathway that produces three different enzymes in the process of creating lactase. So that's what LAC-Z, LAC-Y, and LAC-A represent. The LAC repressor is also, you'll see that upstream a little bit here of the operon for the gene for lactose. So the LAC operon is a negative regulation operon using a repressor protein. Now, lactase does not always have to be present. It is made in kind of low amounts, but for the most part, the repressor binds to the operator to block the transcription and therefore the production of lactase. But when lactose is in high amounts, the repressor, which is produced in an active state and bound to the DNA, right, bound to the operator, blocking transcription, when we get a bunch of lactose in our cells, an inducer molecule, which is a very close relative to lactose, is allolactose. But because of lactose, an inducer binds to the repressor protein. When the inducer binds to the repressor, that repressor gets shut off. It cannot bind to the operator. And if the repressor is not bound, then RNA polymerase can go ahead with transcription. So the LAC operon, again, is a negative control operon, but it is an inducible operon because of that inducer molecule, allolactose, that binds to the repressor, turning the repressor off, allowing the operon to work or to be transcribed. And so here you can see in the image, right, we're not going to worry too much about the cap and the cyclic AMP binding site, so don't worry about that purple guy. But the pink LAC repressor is always made in the active state, and so it binds to the operator when no lactose is present. But as you can see here, when lactose is present, or allolactose, which is our inducer molecule, is present, it binds to that repressor, turning it off, keeping it from binding, right, because it changes the active site. So it can no longer bind, and now RNA polymerase can transcribe the genes for the production of lactase. We're going to skip this part on glucose repression because it's just a little bit more than what we need to worry about at this time, so cross that off on your notes. And let's talk about the trip operon. So the trip operon is involved in the pathway that makes tryptophan. This operon produces tryptophan when there's not enough tryptophan in our cells. So it's kind of usually on, especially, but it will be turned off if you eat high amounts of proteins, or if you eat enough protein, then you're getting tryptophan, and so then it would be turned off. But it's typically expressed then when our tryptophan levels drop. And so the repressor for this operon binds to the operator site. You don't need to worry that it's a helix turn helix protein. We're not going to worry about that. But the trip repressor binds to that operator, which is, again, we already know that by the promoter. So when the trip repressor binds to the operator, that is a negative regulation because we are blocking transcription. Okay, so that repressor will stop the transcription to make tryptophan when tryptophan is present. So it talks about a co-repressor. It says the binding of the repressor to the operator requires a co-repressor. That is tryptophan. So tryptophan basically activates the repressor and binds to it. So when there's enough tryptophan present, that repressor is activated and it will bind to the operator, turning off transcription of the tryptophan gene. However, when your cells use up all that tryptophan, making other proteins or whatever it's doing with that amino acid, when those tryptophan levels start to decrease, the repressor now cannot bind to the operator because tryptophan, that co-repressor is gone. So now it cannot bind there. So if we take a look at this image, right, the TRP repressor is in an inactive state as it gets translated from the regulatory gene. So that tryptophan repressor is kind of in an off state. It cannot bind to the DNA, and so my RNA polymerase here can make tryptophan. So when there is no tryptophan present, then we know we're going to do transcription and make it. However, if we have tryptophan in our cells, you eat some turkey or some kind of protein, that tryptophan binds to my repressor as a co-repressor, and that shape change allows this active site to bind to the DNA. And that binding prevents the RNA polymerase from transcribing this gene. So transcription cannot occur. And there's another image of how the tryptophan repressor works.